Good evening and hi. Today, we'll be presenting a study titled Discovery of Variation in Genes Related to Agronomic Traits by Sequencing the Genome of Cucurbita pepo variety. We are from Grade 7 and our presentation will be delivered by 4 members. So for the case study, it is focused on Cucurbita pepo, which is an economically important cucurbit crop that includes pumpkin, zucchini, and also squash. This species is highly polymorphic, meaning it shows a very wide variation in fruit morphology, texture, and also plant characteristic among cultivars. The traits observed in Cucurbita pepo are strongly influenced by genetic regulations and metabolic pathway. Different genes are involved in controlling plant growth and development, fruit shape, color, and also fruit quality. In addition, metabolic pathway that regulate pigment accumulation also contributes to differences in morphology and appearance of the fruit. However, Cucurbita pepo is susceptible to several important diseases, especially viral infections such as zucchini yellow mosaic virus, cucumber mosaic virus, and also tomato leaf curl New Delhi virus. Besides viral diseases, powdery mildew fungal infection also causes significant reduction in crop yield and also quality. Why is this study is very important? Despite the economic importance of this crop, genetic and genomic study in Cucurbita pepo are still limited compared to other species. The reference genome for Cucurbita pepo only became available in 2018, which opened opportunities for further genomic and diversity study. Understanding the genetic diversity of this species is important because it can help to improve crop quality, identify disease tolerance genes, and also support future breeding programs. With the ability of genomic resources, researchers have all started exploring the molecular basis of disease tolerance in Cucurbita pepo. For example, previous study identified SNP markers on chromosome 1 and chromosome 8 that are associated with tolerance towards ZYMV infections. There are 3 objectives for the case study, which is the first one, to resequence 8 Cucurbita pepo varieties used as founders of a magic population in order to obtain genomic information of the selected cultivars. The second one is to analyze the genetic diversity among the selected cultivars and investigate variation associated with different morphotypes and horticultural traits. And the third one is to identify gene variation related to plant morphology, fruit quality, and also tolerance to viral and fungal diseases.
Let's look at methodology. First, plant material and DNA extraction. This research utilized 8 varieties of Cucurbita pepo consisting of 4 varieties from the zucchini group and 4 from the cucuzza group. Seeds were sown at the Unina greenhouse and genomic DNA was subsequently extracted from leaf tissues using the DNeasy plant mini kit. The quality and quantity of the extracted DNA were verified through agarose gel electrophoresis and nanodrop spectrophotometer. Second, library construction and sequencing. Genomic libraries were constructed from purified DNA using the TrueSeq DNA sample prep kit. Sequencing was performed on the Illumina HiSeq 1500 platform to generate 100 pp pair and reads. This process successfully yielded an average of 40 million raw reads for each variety. Third, data processing and genome mapping. During the pre-processing phase, raw sequencing data were filtered using Trimomatic to remove adapters and poor quality bases. For mapping, the remaining high-quality reads were then aligned to the Cucurbita pepo version 4.1 reference genome using the BWA-MEM software. The resulting files were processed through SAM tools and Picard to manage SAM to BAM conversions to determine number of reads, secondary and mapped reads. 4. Population structure and genetic variability. In genetic relation analysis admixture used admixture Linux 1.3.0 software, 3D visualization data used Curly-Wurly software. A consensus phylogenetic tree was also constructed in Mega 11, Kimura 2-parameter model, and Bootstrap applied to assess confidence. Lastly, SNP and in-depth functional annotation. The biological effect of genomic variants was analyzed using the SNP-EFF program. Pathway analyses were carried out using gene ontology (GO) for gene function classification (KEGG) to identify function and metabolic pathway. mimicry, and MedMem software for biotic stress responses.
For the next part is result. As the table beside show is principal component analysis, which is PCA, was used to evaluate the genetic relationship among the 8 cucurbita pepo variety. The analysis successfully separate the varieties into two major groups, which is Zucchini group and the Cocozilla group. The first principal component, PCI, is explain 38% of the total genetic variation, while the second one is second principal component, PC2, which is explain 80.4%. The variety within the same group cluster closely together, while the white taker separately far from the ascension from the other ascension, which is it show the distinct genetic composition compared to the remaining variety. Okay, for the next part is the second result. As you can see, as the table beside show the phylogenetic tree was constructed to the future analysis the evolutionary relationship among the zucchini varieties. The tree show that the Cocozilla variety clustered together in one branch, while the zucchini variety from another branch. This result supported the PCA analysis and confirmed that variety with similar morphological characteristics also share similar genetic relationship. White taker appear more genetically distant from the other variety due to its complex genetic background and introgression from multiple parental sources. Okay, for the next result is as you can see the table at the right side is genome sequence of the 8 zucchini variety generated an average of 40 million row rate per sample. From this sequencing process, more than 4.7 million genomic variants were identified, including SNP and indel mutation. Among the analyzed variety, white taker contained the highest SNP frequency, suggesting a high level of genetic diversity, indicating that this variety were genetically diverse. In contrast, 381E and 9C8RB show lower SNP variation, indicating that this variety were genetically closer to the reference genome. The presence of large number of SNP demonstrate that strong genetic diversity exists among the zucchini variety analyzed in this study. For the next part, for the table 4 result, several important genes relate to plant morphology and fruit quality were identified in this study. Variation were detected in genes associated with plant growth, leaf development, and fruit shape, also fruit color. Genes involved in the gibberellin pathway were associated with difference in plant growth habit. Homeobox genes were linked to leaf shape development, while muscle and clavata genes were related to fruit morphology and local number. In addition, carotenoid pathway genes were associated with fruit rind color variation. This finding suggests that genetic mutation in this gene may contribute to the visible morphological difference observed among the zucchini variety. For the next part is discussion. The first one is the high number of SNP identified among the zucchini variety demonstrates strong genetic diversity within the population. Genetic diversity is important because it provides a wider range of useful traits that can be utilized in breeding program. The PCA and phylogenetic analysis further confirm that existence of clear genetic separation among the variety. The unique genetic background of white taker explains its higher SNP frequency compared to the accession. The second one is variation in genes related to gibberellin pathway, the leaf development and fruit morphology may explain the difference in plant architecture observed among the variety. Mutation in developmental genes can influence important horticultural traits such as plant height, leaf shape, fruit size, and fruit color. These traits are important in improving crop quality and market value. Last decision is the resistant variety, particularly 381E and 968RB contain several important resistance-related genes. This gene may contribute to the plant's ability to tolerate ZYMV infection and powdery mildew diseases. The identification of SNP-rich chromosome region associated with resistant trait may help future researchers identify candidate genes responsible for disease tolerance.
Hi, my name is Noratira. Next, I will present the comments and evaluation for this study. One major strength of this study is the large-scale genomic analysis. The researcher identified more than 3.8 million SNPs and around 387,000 alleles, showing that the dataset is very comprehensive. Another strength is that multiple cucurbita pepo varieties were used, which show high genetic diversity among the samples. The study also identified candidate genes related to important agronomic traits such as plant growth, leaf shape, fruit shape and color, as well as disease resistance. In addition, the researcher successfully linked genomic variation with rare agriculture traits, making the findings useful for crop improvement and breeding programs. This study is important because it can help improve crop yield, fruit quality, and disease resistance against diseases such as ZYMV and powdery mildew. The findings also support plant breeding program and contribute to food security and sustainable agriculture. However, the study also has several limitations. Most of the analysis was based on computational and bioinformatics approaches. So many candidate genes were predicted but not fully confirmed through laboratory experiments. In addition, the study only focused on a limited number of varieties, which may not fully represent the genetic diversity of Cucurbita pepo. For future recommendation, further laboratory and field experiments should be conducted to validate the function of the identified genes. Researchers should also study more population and apply this finding in real breeding programs to develop a stronger and higher quality crop varieties. In conclusion, the study highlights the importance of genetic variation in crop improvement. The researcher identified genes related to morphology, including plant growth, leaf traits, and fruit characteristics, as well as disease resistance against ZYMV and powdery mildew. Overall, this study provides a valuable genomic resource for zucchini breeding and future crop improvement. And these are the references used for our presentation. That's all from us. Thank you for your attention.